I guess we can start. So welcome to the first competition on the critical assessment of data privacy and protection. And this competition workshop is co-sponsored by iDASH and the collaborating IOM project hosted at Indiana University. So I'm going to say, just in the recent decades, the most exciting scientific breakthrough has happened in human genomic research, right? And with those high throughput and genomic sequencing technique, and the cost for DNA sequencing goes down quickly, and reaching this $1,000 mark. And in the near future, what we can imagine is our DNA sequence can be extracted, attached to our medical record, just so our physician can provide us personalized health care. And also the availability of just large amount of genomic data also provide biomedical research unique opportunity to analyze the data, to identify the markers for those hard to cure disease, to come up with just a new solution. And to sustain this scientific revolution, one of the most important issues here is to make available a huge amount of data, to make such data accessible to biomedical researchers. But just to make this happen, a problem we need to address is how to protect patient privacy. Right? Genomic data carries highly sensitive information. If the data has been leaked to just wrong party, then the patient will be in big trouble. Right? As you can imagine, if insurance company know your susceptibility to certain disease, even if you don't care, maybe they can infer your kids and your relatives susceptibility to certain disease, right? So they will be in big trouble. So a grand challenge we need to address here is how to just make huge amount of genomic data available to the community and make them easy to access. In the meantime, without undermining the privacy protection of patients. So just a few years ago, we thought maybe a simple idea is we just aggregate the data, right? We cannot release individual person's raw data because we already know, just without metadata attached to those raw data sequence. What can still happen is a mapping between the genotype, to phenotype. Although the technique for this purpose is still not there, but we're quickly moving toward this end. So people thought maybe we can aggregate, let's say, hundreds of people DNA together. We just release the frequency of the genetic variations. For example, the frequency of SNP. And just back, let's see, 6 years ago, right? Thomas and Craig and other people published several papers which shows, even with this aggregated data, if the attacker somehow gets the second sample from the patients, they can still determine the presence of the individual inside the aggregate data with high chance. And later on, we show even if you just publish the result of data analysis, for example, suppose you perform a genome-wide association study, you only release the outcome of p-value, I-square, and all this information. There's still chance for the knowledgeable adversary to analyze the data, to really identify the presence of individual inside this, inside clinical study. They could even, recover part of the raw data under some circumstances. So the question is, what are we going to do, right? The whole purpose of research, we're not trying to paranoid people, make people paranoid. We're not trying to prevent the further progress, in the scientific research, in biomedical research, right? And And the purpose, we want to help people to understand the privacy implication of data release, so that we can come up with a better solution to release the data, also safeguard, just privacy of patient. So another solution, just to address this issue, a popular solution is how about we just do data anonymization. We anonymize the raw data or the outcome of just those genome associated studies, right? By adding noise into the data, right? Just the purpose is we want to make data still useful, but without leaking out individual patients' information. But to make this happen, you need to achieve very delicate balance between two ends, right? If we add too much noise, then of course an individual can no longer be identifiable. But in the meantime, the data will become no longer useful, right? If the amount of noise added into the data is insufficient, still you can identify individual. So the whole purpose here is we want to. So the whole purpose of the competition is we want to let the best group, the best research group in the data privacy, genomic data privacy, to bring on their best algorithm to run this algorithm on real genomic data to serve the purpose of real GI study, right? And we want to understand the distance between what can be done today and what needs to be done. What can be done today and what needs to be done, to make the whole technique practical. So Xiaoqian here, we will give you guys more detail about the organization and participation and other information of this competition.
Thanks, Xiaofeng, for the introduction. So let me give you a little bit of history about this iDASH privacy workshop. We have been hosting this event third time in a row since the establishment of iDASH NCBC. And this is a real study, a real impact. This time we decided, different from the previous two sessions we hosted in UCSC, which focused on the methodology foundation. So we decided, why not let's try a task-driven competition and let the researchers present utility and the risk, and let people to judge. So the idea is we try to create this platform, balance the privacy protection and utility, compete different state-of-art mechanisms on real privacy data using human genome information. So we started this preparation about a year ago. Actually, we have this idea and inspired Dr. Luciano Machado, and I talked to our Indiana fellows. So we decided to come up with this competition. After 2 months data preparation, we opened the registration. We waited for the submission of the result, and take about another 2 weeks to evaluate the performance. And today is the day we present all the results. So there's 33 registrations up to now, and we still have a few online attendees which didn't register. And the attendants are from 2 countries, 9 states in the United States. And there are 6 teams participating in this competition, including University of Oklahoma, UT Dallas, McGill University from Canada, Carnegie Mellon University, UT Austin, Indiana University is participating as a baseline. So we are hosting this event for 2 specific tasks. The first one is to privacy preserving data sharing. Basically, when you get the data, and how can you generate a synthetic data set that has a mediated privacy risk while maximize the preserved utility. The second task is to retrieve the key features from the data. So basically, can we do privacy preserving feature selection? So with that said, the brief introduction of today's agenda is we will have Dr. Luciano Machado start with the keynote presentation, and then Haixu, Xiaofeng, and Shuang will set the stage. So discuss about how we set up the mechanism for privacy preserving challenge, and how do we collect data. So that will be about 30 minutes, and then followed by the presentation. the task 2 go first because we have 2 sessions in the morning, and then we leave the 4 sessions to the afternoon to present task 1. And then we will have a panel discussion. After that, we will summarize today's presentation. With that said, I will leave to Dr. Lucila Machado.
Who is displayed.
It's cute.
Right, great. So before anything, I would just say I reviewed some slides from my colleagues here. And I am very glad you all came to San Diego. We apologize for the bad weather we're having, but that will make at least you stay focused in the workshop. So I want to just present a little bit about iDASH and what the project was, which is about data sharing, but how to do that in a way that patients in particular don't get anxious about their data going to people who should not have those data. And it's funded by the NIH, the Office of the Director, and then by the institutes after that, NHLBI, NHGRI, IDFB, National Library of Medicine, and a few others. So our goal is to share access to data and to share access to computation. We do realize that sometimes you cannot put data out there to be shared, to be downloaded, but sometimes you can bring users in to use that data in your center. We want to train the next generation of data scientists, and we have the summer internship as well as some other activities, and provide innovative software, platforms, and infrastructure so that we can share healthcare and biomedical research data while at the same time protecting the privacy of everyone, patients and also of institutions. So with this 3 models of data sharing, what I wanted to essentially put the main points of iDASH, one of which we'll be talking more about today, the data model number 1, where we put the data out there, someone downloads the data and processes at their own place. But I'll talk a little bit about how we're constructing the environment to model 2, in which you come to the center to perform your computations on data that may not leave the center. And a third model, which is how you can use tools and VMs developed in the center to do your computation on your own data without that data living in your own institution. And that is a model that I'll give an example of each one of those 3 as we go along. But one of the things that I wanted to say is the center, so the center for data sharing, in the middle, is based on infrastructure that has been built over time since the beginning of the project. And it's just illustrated here with some of the items that we thought were very important to do. Some items are related to security. They're not related to the other aspects and privacy protection. But it does have redundant firewalls, redundant RSA, two-factor authentication appliances, and it does HIPAA requirements of encryption at rest, in transit, centralized logging of who access what, intrusion detection, proxies and filters, and hardened configurations. So right now, for data model 1, the one that people download the data, we have 3 options. And I would like you to consider those options as you share data sets in the future. One at the top is called known protected health information data sharing option. Does that mean it's always public just because it's known protected health information? No, because as you know, biomedical research can remove identifiers, therefore not be considered protected health information. But still, people don't want to release the data out there. So for that, we have what we call the iDASH repository, and I'll show a little bit of the functionality here today. And also for the protected health information, then you don't even see it from our website, but we have an infrastructure to hold data sets for healthcare, for example, with names of patients, with dates of birth, with medical record numbers, because some research allows that. We have the IRB approvals for that. So there is one repository in the iDASH that I'll show next. And many of you know RADCAP, which is software developed by Vanderbilt University, which we also hold under a very well protected environment to do protected health information exchange. So our two-factor authentication is of two sorts. The licenses are either for the actual physical device or for more popular cell phone, smartphone uses. And it works really well. What you do is when you log into the repository, there is two levels of login with the two-factor in the login for the repository. It's based on open source software that is modified for our particular needs, and it gives access control at the file level. So to upload the data, it's a very simple function. You can upload up to 2.5 gigabytes in one individual file, which is more than many of the other functions or other applications. For example, RADCAP is only 128 megabytes, quite small. And you can select whether it goes to a public or private folder, and also you can select the licenses that go with that particular type of data. To download the data is a very similar process as you do with the Dropbox. It does support compression, and it has, again, much more protection than Dropbox has out there, right? We also have some portions of the data go under the VPN from here, so you have to have special credentials, and therefore you have several levels of security to go with the data, particularly the one that has identifiers, which is not exactly the one that we're talking about here, but as you can see, there are multiple levels. You can start with that, do a whole anonymization procedure on that, and then move the more anonymized data to other levels of security and authorization. So we have two environments. One is the production and development infrastructure that again has all the bells and whistles that you might want to know about. And a new one, which remember is that model that you come to compute in the center, which we're building as a cloud infrastructure. So this is done right now with VMware and vCloud, and is being constructed so that you can do large amounts of computation, private computation with genomes at the iDASH center. So also I wanted to highlight that we have public communities, private communities, and a whole set of access controls and how you set up those communities is all done through software. So once you create a community through this process here, you can select who the users will be, and you can see who modified each file, who had access, and so on. So you can share either private data, public data, and whatever other things you have. There are built-in permission models, so you can select who is the owner, who can edit, and so on and so forth. So again, similar to other software that you've seen, except that it's all under that security infrastructure that I've shown before. In terms of the iDASH RADCAP instance, is again a very popular data collection tool that is of much use around the CTSAs. There are 60 centers in the US that are funded to do clinical and translational research. And it's also under the same infrastructure and has a lot of usages, including what we wanted is to create infrastructure that was available to people outside UCSD. And so, for example, we have some collaborations with Emory University and others as well. So in order to enable this sharing, there is also the legal requirements of it that we cannot overlook. And there are two types of agreements right now that iDASH has developed. One is for the data contribution. So when you want to contribute a data set, you want to say, from now, and I equate that as living your first child at daycare. It's a very anxiety-building process. So you leave your data there, and you keep looking back and see how well it's doing and so on. The second child is easier. And then the third one, you just sit by and then move on. So I would say the first one, because this is a pioneering project, is one that we have more explanation to do. The first users have required more handholding. So we also have a data use agreement. So what happens is you give your data to our data daycare center, and you say only those people can handle my data and can interact with my data. And we are in charge of honoring your requests. So that's for the data user agreement, people who are using those data. So what happens is it was vetted and approved by the lawyers, University of California, Office of the President, and UCSD. And the agreements are downloaded from the iDASH website. So if you have data to donate or to contribute, you go and download this agreement. The paperwork follows that your institution, as you know, you don't own any data. It's usually your institution that owns that data. So the institution has to sign that data contributor agreement, and then you send that agreement to us. Our institution, UCSD, has to sign that. We create accounts in iDASH, and then you can go and set up your community, like I mentioned in before in the MyList repository. So the data contributor agreement is a legal document between your institution and UCSD, and it has a couple of terms, several terms, in which is clearly specified what's the purpose of the data contribution, what the repository host is, iDASH, what it's supposed to do, and what is your role as the collaborator, and you understand all the terms of the agreement. You have to describe the data, and when you're doing that, you have to affirm whether it has protected health information, or if it's a limited data set, which, as you know, does not have certain identifiers, but has some others, such as dates, which are very important for certain types of research. Or if it's a de-identified data, as per HIPAA, which is 18 identifiers, which you probably know very well, or if it has no human data. Then, so once you define this, the type of data, and then you have also to say what's the acceptable use of the data. Who is going to have access to it, and how they can use that data. You essentially define the level of sharing. This is on the data contributor side. So you got your kid and you left there in daycare. And now there are the people who will interact with the data, right? What can they do and what they cannot do. So there are data user agreements. So the user is supposed to read all of this and agree with certain terms, which we highlight here. In terms of purpose, the agreements different from the data contributors because the purpose is a little different, and what users have to do or have to promise that they will do with data under what circumstances. So this is, again, it's not an individual signing, this is the institution of that individual taking responsibility for the use. So this is very common, and as you know, usually what happens is there is one institution who has the data, has to set up multiple data use agreements, and so on. So what we're trying to do with this is that they only set up a contributor agreement, And we, as iDASH, set the data use agreements, serve as a hub for that kind of agreement. Otherwise, you would have all sorts of pairwise interactions or many more signages to occur than by doing this. So then there are additional agreements that data will not be removed or reviewed and adhere to this terms and so on and so forth. So the description of accessible data also, it says what kind of data you are going to access and what you need to do because you're accessing that kind of data. So, for example, if it's protected health information or limited data sets, there are rules, as you know, from HIPAA and from federal regulations that you have to follow, or your institution has to be able to protect data that way. And when it's not a limited data set, so it's when it's fully de-identified data sets, there are also some terms that you have to comply with. So that's what happens. You find the type of data and then you agree with the terms. And the terms, again, might depend on what the data contributor agreement was. So institutions would sign agreements right now from the data contributor agreement side on the left-hand side. There are national institutions. There is currently just one international contributor. And from data users, as you can imagine, there are more. There are more users accessing some of the data sets, and they're listed on the right-hand side, including some international institutions. So this is all to say that iDASH is not the technology, the infrastructure, but it has also go with the policy behind it, because otherwise the data models for sharing cannot actually work. So you're here for the biometrics and protected health information steps, and remove some of those identifiers. And even when it's de-identified by HIPAA, you all here are researchers who know how to re-identify that data. So our purpose here is to, like Xiaoshen mentioned, make that harder while not destroying the usefulness of the data. So biometrics are protected health information, and they require HIPAA, which is the Health Insurance Portability and Accountability Act, which is the best item that I had for HIPAA is right there. And so what does that mean? That means if biometrics require our PHI requires HIPAA, it follows that biometrics require the HIPAA protections, correct? I don't think anyone would here deny that that's the case. And that being the case, what you have to do is to imagine now you get all these genomes yourselves, and there are many public ones right now. So what happens when you switch from this to this? You have a much better biometric than a fingerprint, right? You have the ultimate biometric in your hands. So that's where, again, not only you cannot leave this data unprotected, but also you need to make sure you preserve some value into it. So in task 1 today, you will be talking about how to protect that data, and also you will be talking about the value of the aggregates, or how much the aggregates leak some information about individuals. So I just wanted to quickly say that people usually don't understand, that's how you do here, why genomes, if you take the name and if you take the date of birth and medical record number, they are at risk for the individuals who donated that data. And it's hard to explain sometimes, but imagine the situation in which you have a study of HIV treatment, 2 arms, standard of care, and the new drug. So all patients in that study are HIV positive, and they are receiving 2 kinds of drugs, and you have the genome and you have the identified data regarding their outcomes and other things that are happening. So what happens if you are interested in knowing whether your neighbor is HIV positive or not? Essentially, you can get, and you know how to do that now, get the DNA of your neighbor and check against the data set, and any other data set that is public out there, right? So by uniquely identifying your neighbor in a data set that is about a certain disease, a certain condition, and has other items there, you're essentially finding the information you wanted. And one, the patient who contributed to the study does not necessarily know is at risk. Because who would imagine, 10 years ago, that someone could sequence your DNA and check against something else, right? So times evolved, and things that were true 10 years ago, they are no longer true now. And the threats that were not true 10 years ago, they might be true now. And that's where a lot of this has been evolving, and with DNA as well. Think about it, you're not only, let's say it's not HIV, it's a chronic condition that this database is containing. So you're revealing about information about yourself, and you're revealing information about family members as well, who did not decide whether they wanted to be in the study or not wanted to be in the study, or consented or not consented. So that's where there's a lot of movement going about this right now, including here in California. We have across the state very large biorepositories. And these biorepositories were collected in an era in which the blood would be measured for cholesterol, and this would be measured for something else. It happens that now the DNA can be measured or can be seen. So are we allowed to use that for genetic research or not? And there's clearly debate around that. But to prevent that situation in which I said you get your neighbor's DNA and you match against existing databases, this is why this law is still being discussed. It hasn't passed yet, and there's a very large lobby against it because the law would prohibit from collecting, storing, analyzing, and disclosing genetic information without the written authorization of the individual. And there are many institutions that claim, and very much it is true, that it's highly impractical to go back and reconsent everyone who donated to large biosample repositories. So it is something that is food for thought. You can see the advantages and disadvantages on both sides. And here I just listed a couple of things that it goes about, but essentially what it doesn't want is the ability for anyone to go and get your hair sample or get other things and do it without your knowledge. It's like collecting your fingerprints without you knowing that it's happening. Given that fingerprints reveal very little information about your health, it can reveal a little bit, but not as much as DNA can. So here is the whole thing about individuals' authorization. And this is a case that came out last summer that I thought it was interesting. From Boston, and this was a serial killer. It was a victim who wasn't sure, family wasn't sure, he was in the late 50s, of whether the woman was a victim of this particular serial killer or not. But in order to take the body out and do the DNA off the serial killer, it costs a lot of money. So what they did instead, they took authorities need to match through DNA taken from a water bottle thrown away from the nephew of this serial killer without the person knowing. And they, the family got very upset about that because it's essentially in this case that the California law is trying to prevent that no one takes your DNA and sequences and uses for whatever purposes. So this was just to illustrate that this can happen, and it's perhaps happening without us knowing. And why does it happen? Because I believe there are holes in the way the consents are done now, and the language is very ambiguous about that. So, for example, this is an example from one institution, and it says your medical information can be used for the purposes of quality improvement, patient safety, education, and the fluid cells and other specimens being used and shared with research. So, of course, under regulations. And this is when you go for care. So you go, you have a cut, or you have a fracture, you go to the emergency room, you do sign things like this, which probably none of you read, but you have sign. So you also saying that this sample will be shared, or the data will be shared in accordance with the federal law, and then in a way that goes with the notice of privacy practices. Then it's also another document that is not exactly given to you at the point, but is available for consultation. And one of this notes says, so I just want to put this translation of this book into Portuguese. It's a bestseller book. I hope you read it. It's very interesting. And also because about this time last year, there was a lot of controversy when someone put the sequence of the HeLa cells online, and then they had to take it out immediately. And again, it's the same thing. The cells, in this case, were not even collected with the consent of the patient or the family, and then they went out there, they were commercialized. And then someone thought, well, the cells are out there, I'm doing a service to everybody if I sequence them and I publish them, which I would say is very a good thought, and it's an altruistic thought. I put my money to sequence those cells, and now I'll make it useful for others. But there are decisions of privacy from the family that need to be dealt with before. So sometimes you think you're doing a good thing, but maybe you're not, or you have to verify it before. So here is that notice of private practices that I told you about, that I received now that the insurance changed, but like everyone else was reading in detail. This is not mine, by the way, but it's very similar. So the data and specimens will only be shared if you note your willingness to participate by signing a consent form. When approved through a special review process, other studies may be performed using your medical information without requiring your consent. And in those, it's weighted the practicality of getting the consent or not. So the biosamples, for example, at this point, the large repositories of biosamples, it's considered impractical to go and obtain the consent of the patient. So this is currently one such case that could go through special review process. And there are some questions about data sharing at the midpoint. I always like to see if people are paying attention and whether they understand some issues. Internal Review Board, IRB, is something that is required to do certain manipulations with the data mining. Which types require IRB approval? Does quality improvement in the hospital and the clinic require IRB approval? Who votes for yes? So no, it doesn't require, thank you, you were on top of it. If you want to publish that quality improvement, does it require IRB approval? Who thinks it does? So all wrong. Oh, one person, right? Very subtle. It does require, so you have to get it back, retroactive IRB approval if you want to. If you found something interesting, quality improvement in your hospital that one other hospitals you know, you need to get IRB approval because it's considered research from that point onwards, because it's disseminated outside your own institution. Public health also does not require IRB approval, as you know. and which types of data sets could be used for research without patient consent. Who votes for A? Yeah, A can. So HIPAA de-identified, 18 identifiers out, can be used. Doesn't mean it cannot be re-identified, right, easily. Limited data sets. And yes or no? No, because you submit to IRB, and if your IRB is kind of, I would say average, you would say no, it's safe enough, you don't need to protect. What about identified data? Require IRB or no? Most of the times, yes. There are a few circumstances in which might not be practical to obtain. So like emergency department studies, not always can have the consent of the person of the family. So that's our special case. What are the implications of improper disclosure, reputation, money, time loss, and privacy loss? Can a healthcare institution honor individualized patient preferences? Not so far, and this is what we are trying to do here in iDASH, is to shift control from the healthcare institutions that currently decide all of that. Who shares what with whom, according to IRBs and others. So the way we're trying to do is to make it so that the patient can have a little say or a large say into what can be shared. And it deals with everything that I talked about before, policy. It deals with technology, and it deals with a certain place to have those consent preferences stored, in a safe storage of those consents. And it has to negotiate with each healthcare institution of what can be shared or not on an individual basis. So it's not necessarily a simple process. And that's why we are doing a pilot right now with our clinical data warehouse, in which we will get preferences from 100 individuals and implement that when we do data sharing and understand the patients are more satisfied with that, whether they don't change any preferences, anyways, or things of that nature. So we have been looking into this patient-centered data sharing aspects for a number of years now, and we are evolving. We have 3 projects right now in which we deal with informed consent in different populations. So they just started about 6 months ago, and they deal with different aspects of data sharing, one of which might be the one that you're most familiar with, which is sharing data across different institutions and downloading, uploading data sets. Another one is, like I said, to create our infrastructure so people can create this blueprint that you can do certain analysis on certain types of data without moving them out of the institution. So this is the cloud that we're constructing. And another one has to deal with who is obtaining that consent. So if you've ever been to a clinic, sometimes the consent is obtained in the most unfortunate time, when you're most stressed, when things are, all you need is the care, you're not caring about research at all. And then who gets that consent is very important. If it is the doctor right there who is asking you, you might be reluctant to say no. If it is a research assistant on the side, you might be more comfortable saying no, I'm not going to participate, or in my case, I've been through this situation, I don't want my son to participate in this, I can't see the benefit. Or okay, it's fine. It won't delay care, because that's one thing that you're always thinking. And sometimes it's inconvenient, but the benefits, the knowledge that comes about it might be very interesting. So just an anecdote, when I went, my son was probably 8. And he had this abdominal pain, which looked completely like appendicitis. You could take him to the emergency department and have and I'll see what happens. And the resident comes, oh, we're doing this study about CTs instead of going directly to surgery. Would you like him to participate? You're calling the surgeon, you're still doing other things. And what you have to do for the CT is just prepare the intestines, and then it's the same preparation that goes for surgery. I said, fine, if it does mean that you won't have the surgery because they will discover something in the image, that's pretty good. Who wants surgery anyway? So I consented to that, and the kid goes and has an enema to prepare for the CT. And then he goes to the bathroom. I said, Mom is not hurting you. And I said, wow, are you sure? Because it was typical appendicitis. The nurse had already met with another one. Monday, this is appendicitis, and it won't come. And the research just saved the day, because after the enema, he did not have the abdominal pain anymore. He didn't even have the CT in that case, but that's all right. So research does help. That's the bottom line here. So as I said, that's the other study of whether the research assistant or the surgeon asking for consent changes what happens in terms of the patient wanting to participate. And finally, I want to say that we had a lot of help for this research and for the technical aspects of privacy technology by having trainees in our program. So last year we had a lot of projects, and everyone was one of the participants here, and I'm glad to see you back to the workshop, in which we had different aspects of this in the study. So again, well, the internship applications are due March 27th, so there's still a lot of time to prepare them and send to us. And again, it could be in one of the three different data sharing models or policy and other things that we are dealing with. So finally, just to go about the last model, the model number 3, virtualization and distributed computing. The reason I wanted to say something about it is because we're embarking on a project right now that will use exactly that type of computing. And it will use data from what we call UC-REX, Research Exchange, of the 5 medical centers in California. That's 1/3 of the California population. And it's for patient safety, surveillance, quality improvement, and translational research. So the idea is that each one of the medical centers has its own clinical data warehouse that we can consult for certain aspects. Currently, we only do count queries. Count queries just say patients with this and this. There are 3 in LA, 5 in San Francisco, 2 in San Diego, 1 in Irvine. So do you think that's privacy preserving or not? Is it? I'm just giving numbers back. 3 patients here, 2 patients there. So, and I'm allowing you to query that several times. So it isn't, right? And we have examples of that even with our own data warehouse. Of someone wanting to find certain patients and querying a lot and modifying the query so that you know who you want. So that's one of the things that, in order to mitigate that, we have very loose kind of rules. We're just saying no patients, no counts below 10 will be returned. So that's what we do right now. And no counts for patients over 89 will be returned. Because as you know, 89 is the HIPAA threshold for being able to declare age in an de-identified data set. So as you can tell, that's not very rigorous, but it's restricted individuals from the UC system. And we know who queried what, how many times. But there is a mechanism to try to prevent the sequential querying, unless you ask someone else to query it or something a little different, right? So, but it's all around the same topic. So that's one thing that is happening right now. So future steps, we do want to share individual level data, and we want to use iDASH as the repository to do that for protected health information included, because we have what I showed in the beginning, and to do cross-institutional analytics. So one of the use cases that we used was this one of warfarin and dabigatran, which are drugs about anticoagulation. And we wanted to know if the new one, dabigatran, was as safe as warfarin, or better, as clinical trials had indicated. So in order to do that, you want to know what the major minor bleeding rates for patients on these drugs are, as well as when they're not anticoagulated enough and they have strokes, they have other events. So what you do to do that, and we had three different, this is a different, it was not UC-REx, this was a VA UCSD in Los Angeles clinics study about if we could find this through counts alone. But counts are not sufficient. So what we came about as well was with this distributed regression models, because you want to adjust for confounders, right? You just don't want to say this many cases of major bleeding, but given that your cases were all more severe than some others. So you have to do multivariate adjustments across the 5 systems. So folks from this group came up with this distributed regression model in which you can calculate a regression model distributed way in the same way as if you had collected all data centrally. So in this case, data never move from where they are. You calculate the coefficients there and you combine them centrally. And then you guys that are privacy folks, and you will say, well, but the return of those coefficients might not be privacy preserving. And that is true. That's why you're all here, and you can discuss how to do distributed regression, which is privacy preserving, but it's not model killing, as I say, because you introduce so much noise into the coefficients and other things that the model becomes useless many times. So this is one of the things, again, about that distributed model. The other one that I wanted to end up with was this new initiative, which we call P-Scanner, Patient-Centered Scalable National Network for Effectiveness Research. And it's part of the PCORI, this is the Patient-Centered Outcomes Research Institute initiative, to create a national network for comparative effectiveness research. So the goal here is to network the whole country in order to learn from electronic healthcare data as well as other data what is happening and how to provide better care to the patients by having more data. You're data mining people, you know you need a whole lot of data in order to extract real signal. So what we decided to do here is to merge the efforts of the UC-REx system on our 12 million patients with the National VA Clinical Data Warehouse, which is centralized in Austin, actually, the data center there, and it's 9 million patients, plus federally qualified healthcare centers that is safety net clinics in LA, and that added another component to this network. So overall, about 21 million people whose electronic health record data extracted into clinical data warehouses we would be able to access. And then connect that with 10 other such networks that exist around the country. Very interesting project. One network is the whole New York City network together. The other one is the whole state of Louisiana. The other one has to do with the safety net clinics across the country. And one has to do with pediatrics. So a whole lot of diversity in there. And estimated about 100 million patients have some duplications granted, but 100 million patients that could be covered by this network. and many of them are right here, so the University of California patients. So a lot of that, but of course a lot of noise in the data, a lot of issues regarding how we harmonize the data and how we get to where we need to get. So I wanted to end up with this because it's a project that's just starting this month, practically, and that has a lot of issues regarding privacy as well. It is constructed as a distributed research network at the moment, but within the University of California, we have intentions to also use some forms of centralized models for certain kinds of studies when it does make sense. So that, and also the distributed computation, as you know, certain algorithms will not lend themselves for being distributed in that fashion. Also, as part of these national networks, there are what they call patient-powered research networks. And this is collections of patients who decided they wanted to have a registry or they wanted to have a database so that more knowledge could be derived from patients with rare conditions or patients with they are so engaged that they created their own data collection. So that is again another side of this, that you have what I mentioned before, is patients very, very much willing to share the data in a private manner in a way that advances research. So with that, I'm out of time, and I just wanted to ask if you have any particular questions. This is very general. You get into the nitty-gritty of the competition and of the anonymization as presented with us very soon. But this is to give an overview of iDASH and how this component is very important, because we will, again, always have that model of sharing the data themselves, downloading and uploading. So with that, I'll close here. Any questions?
Yeah, actually, I have a question. I very much like the idea to consider genome as very similar measure as a biometric, and consider them in the same way as how biometric was handled in my question is, there are also some slight difference between genomic information and other biometric information. Basically, genomic information is very fragmented, right? If you look at like fingerprint, it's a single fingerprint. Genomics break down into many different regions. How to define the base and the privacy risk? If you consider whole genome, that's one level of risk. If you consider a single gene taken from a genome, or particular genome loci from the whole genome, that's a different thing. And also there are also single gene tests, right, or multiple gene tests. That's not do whole genome sequencing, but only focus on one gene or multiple gene. Should that be also considered as genome sequencing if you consider genome information as similar as biometric information, then everything will be covered. Even a single gene test would be also violate somehow the measurement of some biometric.
Yeah, and that's why technology solutions, technical solutions are not just it, right?
Right.
That's why
some policies, right,
need to exist, and that's why BBGAP is under some strict access controls as it should be, because of the combination of steps we'll review. But it's the same thing, actually. It's kind of cheaper,
right,
if you just do an array or a SNP array, then you list everything. So the risk will not go away. The need for sharing will not go away. So how can we combine in a way that it's fair? All the risks are declared, they are understood, and they're considered, not to outweigh the benefits of studying all this. So I think it's upon us to show the clear benefits. And I think there were some already, but perhaps not as dramatic as a bedside saving life kind of story. But they are out there. So a lot of issues are about education of the benefits and of the, being upfront that the risks exist, but they can be mitigated, they can be controlled.
I was still wondering whether or not there's actually a practical limit there, because you mentioned that iDASH has the policy to define some study data at the individual level as non-PHI, which is basically based on HIPAA-defined variables.
Right.
And the other thing is the individual genotyping information. Clearly, that's almost like a signature. However, the vast variety of type of data we are starting to collect. For example, laboratory test. If a patient has been treated at a hospital over 10 years, you probably have done a lot of blood work. And you can reasonably make an argument that theoretically, if you know individual patients' blood work over a period, that might be some very interesting identifiable profiling approach.
Okay.
In that case, how do you define a privacy in theory and in practically everything can be, identifiable?
I agree with you.
So where's that?
Well, then again, that's where being upfront about it is more important than swimming under the rug and saying there are no risks, because there are. Dates, for example, then people shift dates, right? Because if I know my neighbor went to the hospital this day, I can check against age and against something else, and I figure out in the data set that has diagnosis what my neighbor went to the clinic for, right? And then people shift dates. But it's a systematic shift because you want to do the study in a way that you shifted whatever 3 months ahead or something like that. Then you might lose, seasonality, right? Certainly, well, if you shift too much, and you might lose a lot of other information. So it's exactly the trade-off between trying to, difficult the job of the bad guy and providing data that are useful. So many people say, well, then don't even try to mask or do this. Just do signed agreements. If whoever breaks the law, find an enforcement mechanism. Because perhaps we are destroying the quality of the data to pretend we are fully protecting against risk, which you all know we're not, right? Mathematically. So it's where the policy probably takes precedence and becomes more of a reality or a practical solution than only technical. So ideally, I believe you combine the two. So my idea of things is you might have a public data set out there. It's not that useful, but it gets you started on something. And if you really think you have a good thing, then you apply for permissions and you do a paperwork. And then you get the real thing. In which case you can say, oh, well, now I can see that indeed when I was investigating is proven true in a true data set, or the other way around. But I think that these technologies that we were talking about is essentially to teeter where you place that data. On the public, completely public, there's certain kind of access control that is loose, or under very strict legal agreements in which it is much more controlled. So I think there's value for this, that's what I'm saying. But if it's not tied with policy, then it's more of an exercise.
Good question. This is much less serious than the other two questions. It's about the summer internship. So, do the students get
serious too?
Oh, that is.
It is.
So, do the students get to choose an advisor before the program starts, or the advisors are assigned to the students, or it's like a mutual way?
When you do the application, you do declare your interest,
right, yeah.
and even your bio, your CV might indicate certain areas more than others, because we try to pair up with the privacy we're already doing, and also with the skills. Some are more programming oriented and algorithm oriented, and some are more surveys and other things. So it does, and it depends on availability of the faculty. But we work in teams, so in a way, it might be an official advisor, but everyone is involved.
Do the students and teachers meet on the daily basis or?
Not daily basis for most. Not because we didn't want to, but sometimes time is limited. But yeah, as necessary, it's very short, 10 weeks. So we do have to meet frequently. And sometimes it is a daily basis, not formally scheduled, so, but you know, someone stops and asks a question. But highly interesting experience on both ends, because we had very good success with papers that came out of it, and also people who joined us, like Schwann's postdoc here came as a summer first group of summer interns, I believe, several years ago. So, a lot of interesting things.
All right. So my name is Haishu Tan. I'm here just to talk to you how we designed the two challenge tasks for this challenge, and I think Xiaofu and Sean will talk about the result. So as Xiaofu and Xiaoqing already mentioned, the idea to have this challenge is to want to see what's the current status of the data preserving, data privacy preserving techniques for application of those things actually preserve privacy risks on genomic data. So we designed two tasks. The first task we want to test in the case like if you want to share to public a set of genomic data. And in this challenge, we focus only on the SNP of low frequency on a set of SNPs, those kind of data. So the first task is we want to mimics the situation. A particular data owner want to share to the public a set of SNP, low frequency on a set of SNPs. And of course, in this case, you cannot share original data, but we want to test the current status of the data privacy preserving technique to add noise, basically, to anonymize the data in a way that the original patient, individuals who contribute to the data cannot be re-identified from the shared data anymore. And the second is the second challenge is slightly different. We basically try to test the technique to select features from a set of SNPs, let's say thousands or millions SNPs, to try to report only the top significant SNP under certain statistical analysis of the data. So the idea is, let's say if I'm a user, I'm interested only the top significant SNP, let's say top 10 or top 5 significant SNPs that are most interesting for me. Then can I just can a particular privacy preserving technique apply on the data so that I can report you the top 5 or top 10 that are interested, but of course there's some data manipulation has been performed, so they're not exactly the same 5 or same top will report you, but a significant number of proportion of the correct true top 5 will be reported still, so that you still can utilize the result in a certain way. So these are the two challenges. But the idea we want to design the concept we want to design the challenges we want to mimic as possible as real as possible the genomic analysis. So we want to basically select the real genomic data. So we use the two different projects. That fortunately or unfortunately, I don't know, but they are publicly available right now. So we don't take any risk. We basically use one of the projects as a HAPMAP project that's basically funded by NIH. There are 174 controls from European population. So the SNP price of this, I think, between 500,000 to 1 million SNPs have been provided on these individuals, and this data were publicly available through the HAPMAP project. And also we consider in the beginning, we want to also select some kind of patients, some type of patient data with some kind of disease disease status. And so we look at the so-called personal genomics project, which were conducted on thousands. I think currently there are about 3,000 people signed on this project. So they basically give up all their privacy, personal privacy, genome privacy. So their data has been publicly released through this so-called personal genomics organization. I think currently there are over 1,000 different data sets has been released through this project website. But after we download all this, there are all kinds of problems with this public data set, and we can only find about 200 cases from this project has nearly complete genomic, whole genome information available. And actually, I should mention in the beginning, if we do not filter, we can actually collect about 411 participants on like 29 million SNPs that we provide. But since we have to match this PGP data with the so-called control from the HapMap data, so we can only collect the data from those SNPs that has been commonly profiled in both data sets, and also we need to consider those individuals with nearly 90%. 90,000 valid genome has been provided across the genome. So in the end, we can only have about 200 patients, 200 so-called patients. Actually, they are participants from the PGP project we use, and there are only about 100,000 SNPs in the genome-wide assay. Their genome types has been assayed on the genome-wide. Also, even for these 200 people, there are always a small, very small fraction of SNPs has missing value. So we use the phasing algorithm, the fast phase algorithm, to fill in the missing values. So then the data we share with the challenge participant will have a full SNP profile across 100,000 SNPs in the whole genome. So the first challenge, as I said, is try to test the algorithm for privacy preserved data sharing. So to this end, we develop some like baseline algorithm to see how much we can do probably. So we can select a reasonable size of data to be shared through the challenge. So in the end, actually, you might in the afternoon we'll talk about the method we use as a baseline from Yale University. But in the end, we choose these two different data sets. The first data set consists of 300 SNPs across about 500,000 base pair in the chromosome 2 of the human genome. And the second data set consists of about 610 SNPs across about 1 million base pair in the genome on chromosome 10. So we choose these two data sets based according to two different criteria. First, we want to make sure that each of these data contain at least a significant number of some number of significant SNPs under case control associative test, and we use, as already shown in the later slide, we use chi-square test for this purpose. Essentially, we have at least like 5 to 10 or 20 significant SNP under different thresholds of a chi-square test, showing significant difference between the case group, which consists of 201 individuals from the PGP data, and also 174 control group, control individuals from the HapMap project. And also, we want to, for each of data, we want to make sure that they have high risk on re-identification. Basically, we can identify a substantial number of individuals from the raw data. So otherwise, there's no reason to protect this data anymore. So we basically want to make sure, first, they have, if we share the original data, it has high re-identification risk. And second, indeed, this data can be used as testing data for the case control association test. So, the goal, of course, for task one is to understand the privacy utility balance. how they can achieve when we share we publicly share the SNP data after some normalization technique. And the utility will be tested on the chi-square association test, and we basically will count the number of SNPs under different thresholds using the chi-square test. And then also we want to make sure the privacy has been appropriately protected by different methods through the anonymization technique. So we basically will, for the privacy protection, we test using this likelihood ratio test, which is the strongest replication statistical test that is known for now. So for each anonymized data submitted by the participant group of this challenge, we will test the likelihood ratio. We apply the likelihood ratio test as we implement it. So we implement an online tool for the participant also to test the privacy risk in their noise added data on the website. So before they submit their final results, they can also test the risk thing. So we want to make sure the risk has been mitigated. So the likelihood ratio test briefly is a statistical method to test a particular individual to be present in the case group or not. So the result of the likelihood ratio test can be shown as a figure like on the right. So the left panel of the graph shows the statistical value of each individual in the case group, and the right is the individual level of a test group that consists of individuals that are not in the case group but have very similar genetic structure as the case individuals. So from this graph, you can see clearly there's risks because the individuals in the case group, a lot of them have significantly high statistical values from this likelihood ratio test than the test group. So you know this person are very highly likely to be present in the case group than a random chance. By random chance, I mean if I take like 5% false positive rate, I will got about 5% false positive identification. And if I got many more individuals re-identified from the case groups, we know that the risk is higher or sometimes it's very high if we don't do any data anonymization. And also, we will use the chi-square test to see which of the SNP is significantly associated with the case and control individuals. And for these two data set, we used for the challenge task 1. The data set 1, we have at least a number, I think about 10 some SNPs that above the top 5 basically have very high significant value. So the y-axis here is a negative log value of the p-value, and the chi-square statistic. You see at least the top ones have a very high significance, like 10 to the power negative 9, negative 10. And the second one also have a few of them have high significance value. So we expect to see after data anonymization, this highly significant SNP will be preserved. They remain to be highly significant, even though the risks has been mitigated after data anonymization. So this is the kind of data we shared with the participants. So this the first line represents the IDs of the individuals in the case group and the control group. And each following that, each line represents the genotype of each individual on a particular SNP. So start from a SNP ID. And the second task the second task of the challenge, we try to see if we can select the correct most significant SNP among the again a statistical analysis of the data. So for in our case will be again the chi-square test on the case control association analysis. So in this case, we prepared two data. The second data essentially is a whole set of 100,000 SNPs. Okay, on the entire genome, and you can see these SNPs are quite widely distributed across the whole genome, across all 22 chromosomes. So So, but we hold this whole data set as for our testing purpose. We didn't share the second data to the user, but we share a subset of the SNPs, about 5,000 SNPs, across the entire genome of 22 chromosomes, and we release this data to the participants so that the participant group can train their algorithm, test the algorithm on this data set and give us the best algorithm they can implement. And we will test on the second data set too. And again, the utility will be evaluated, say, among the top K, for example, K equal to 1, 2, or 1, 5, or 10, or 20, among the top 20 most significant, top K most significant SNPs reported by each of the participant algorithm. We want to see how many of them are actually the real top most significant SNPs on the chi-square under the chi-square test on the original data. So we, of course, in the real case, we don't accept all the K are the real ones, but we accept a high proportion of them are the real ones. So the higher the proportion, or the higher number of true top K significant SNP can be required by algorithm, we consider this the better algorithm. And the privacy protection for this task, we use a higher standard basic differential privacy with a preset budget epsilon equal to 1.0, which means that all the submitted algorithm will have to follow the standard differential privacy budget epsilon equal to 1.0, so that they can be compared fairly under the same level of privacy protection standard. And I should mention differential privacy is a very high standard for privacy protection. It does not rely on any reference group as the other the likelihood ratio test as we use as task one. So so we think that this standard is quite high. So, but we want to still see if the current technique can select as many as possible the correct top K significant SNP and after data anonymization. And this is a kind of the data set we share with the participant of the second task. So again, the first group, the first line shows all the IDs of the participant, of the individuals in the case and control group, and then each SNP, the genotype of all the participants in each SNPs. On each SNPs was separated in separate paragraph, and also the major and minor allele frequencies and the p-values calculated using the chi-square association test was also shared for this task, so that each participant can compare their result with this result to see if they can get a better and good performance on the data set. I should mention, I should reemphasize again, we only share with the participant with the same first data set with only 5,000 SNPs, and we test, but after we receive all the participants' algorithm, we actually retested on the second data set consisting of 100,000 SNPs across the whole genome. I think the next Sean will tell us a little bit about one type of visualization software and others implemented for visualize the results. Yes.
Yeah. Even when top significant SNPs are preserved, if I as a researcher's point of view, if I lose the odds ratio, still significant from 2.5 down to like 1.1, then I see it as a loss in utility. Do you measure?
Right. So we don't measure this right now. And actually, to be honest, for the second task, we didn't even ask the participant to tell us what's the significance level. The only result we expect the participant to tell is the top K. What are those SNPs? Top K SNP. We didn't evaluate the SNP we didn't ask them to submit to report the significance level of the second task. But if you want to test the significance, but as I said, but you can see that the significance itself is, if you know the association test, okay, the significance level is very much associated. It's actually one-to-one correspondent to the allele frequency. Essentially, if you know the p-value, you can derive the allele frequency. Okay, so that means you also need to go back to like the task one. You need to manipulate the allele frequency so that so that you can preserve the utility preserved essentially is the number of noise added to the allele frequency. Right? Let's go back slightly to the challenge one.
So your answer is I don't have to worry about odds ratio. Changes in odds ratio.
Odds ratio essentially is related to the allele frequency, right? That's basically you need to check the allele frequency to see how much odds ratio you can preserve. All right.
Hello everyone, I will give a brief introduction on the widget service. Their widgets stand for Web Interface for Dynamic Genome Privacy Evaluation. Here's a screenshot of our widget service. They summarize some key features of widgets in this slide. First thing, widgets, user can upload their results for dynamic evaluation on data utility and privacy within their browser. Widgets provide both graphical outputs and actual summary with customizable settings. Second, user can compare and share results with other researchers in widgets. I will walk you through with some demos of our widget service. Let's start with the demo for task one. In widgets, user can select a preloaded result through the drop-down menu. Then the first 10 rows of the result will be shown in the right table, where user can scroll horizontally to view more columns in the first Manage tab. The user can also upload new results in this demo. A CVS file was uploaded through the Choose File button. When uploading is completed, the new result will be shown on this page. Users are able to change result name by clicking the Rename Result button. User can also edit or add data description. For example, a user can add results for chromosome 2 with privacy value equals to 1 in this result. Moreover, user can download or copy the dataset. In this demo, we downloaded a dataset in CSV format, which can be opened in Excel. User's computer. Finally, user can remove a selected dataset from memory by clicking the Remove Result button. In the View tab, user can find details about the result. User can select a list of SNPs to start it or specify criteria to query a sub-dataset. Moreover, user can change number of records per page or browsing more data through the navigation button. A user can evaluate utility over multiple methods with different functions and p-value thresholds. For example, candidate functions can be positive, negative, true positive, true negative, and so on. Besides, the p-value threshold can be changed from 0.05 to 10 to the minus 15. Given those selected criteria, the corresponding result will be summarized on the right-hand side panel, such as the mean, median, standard deviation of those selected functions. A user can also scroll down the web page to browse more results for each setting for different results of different methods. Widgets can compare the selected method in box plot for p-value of 0.05. McGill's method shows the best accuracy in chromosome 10. The user can also select a box plot of multiple functions with a given method of a p-value. Here is a box plot of multiple p-values for a given method and a function. We can see the accuracy increase as the p-value decrease in this example. We evaluate privacy risk using likelihood ratio test based on a given significant level. User can compare multiple results at a time.
where the identified and non-identified individual are indicated by different markers and likelihood ratio test. User can also change the significant level in the studies, where any changes will be automatically shown in the plots. Here is the demo for our task 2. In task 2 page, user can compare different methods for given k value. Here k is a threshold indicating the top k most significant SNPs. In this demo, we have 3 different methods to compare, which use the same privacy protection standard. This is the epsilon equals to 1 differential privacy. By changing k value, a widget will generate different box plots, which illustrate the number of SNPs identified by different methods in this case. A second, user can check result of a method over different k values. Widgets will automatically generate a box plot based on user selection. Here, the box plot shows the number of SNPs identified and the different targeting k value for given method user selected. User can also choose different method to solve the result. In the help page, user can find a short description of this project, a list of team members, and we also provide a demo video for users to better understand video service. So I will hand over to Xiaofeng to present the participants and the results.
So just as I mentioned at the beginning of the, beginning of the workshop, the whole purpose of the competition is we want to understand what we can do today, and and what needs to be done, right? We want to understand this distance. So just for this purpose, we invite a group of team, which represent the state of art. All these team, come from a very reputable researcher in privacy protection. Some of them is specialized in genomic privacy, while other parties is well-recognized expert in data privacy in general. Right? So basically, for task 1, we have these 4 teams: University of Oklahoma and UT Dallas, McGill, and we Indiana University also participated, but just as baseline. And when the party will come up with the data, so it's kind of conflict interest. If we will also come here and participate in the competition forward. In the task 2, we have 2 groups, one from UT Austin, the other from CMU. Right? So now the show time, let's see what's the result. And for the first task, basically just as Haishu introduced, and we are trying to release, just aggregate the data, and but we cannot release the data as is, right? We need to add noise to protect patients' privacy. But still, we're trying to make the data useful, and and the individual group, they are expected to, using the released, noise added, released, and those aggregate data, add noise into the data, then run, a chi-square test to find out, the utility still lacking the data and the level of privacy protection. So, just as I mentioned, as Haishu mentioned, we are working on 2 data sets. The first data set contains 311 SNPs, and the second data set, contains 610 SNPs. And we choose cutoff values to determine the importance of the SNPs. And we use 3 cutoff value. The first is 0.05, the second is 1,000, and the third is 10 to the minus 5. And we also using chi-square. We also using the likelihood ratio test to compute the identification rate. So we have this set of identification rates, 0.05, 0.03, and we have different set of identification rate, and based on the the data, the anonymized data generated by different groups. And the identification rate has been computed on a confidence level of 0.95. That is, we have 5% of false positive. So in the case, if you have just an identification rate of 0.05, it means nothing, right? That means you can identify 5 people out of 100, but you have five false positives, right? So you have no confidence whatsoever for whatever you identify, right? So these are the results here. And we, just at IU group, we perform, just two analysis. And, just the first is SNP-based noise adding. It's a very pessimistic approach. Basically, we assume each SNP is independent from the other SNP, right? So add noise to individual SNP and trying to summarize the identification rate. And so, just as you can see here, just at 0.05, identification rate, what we can get here is, just using the SNP-based approach, and at the significant rate of 0.05, right? Originally they're supposed to be 22 significant SNP, and even use this baseline, which is the most naive approach, we can identify 19, which sounds very good, right? But the problem is, and what has been reported by this approach actually is 263. It reported 263 important SNP, but among them, only 19 is true positive. That is indeed the SNP which is important. And the others are false positive. The false positive introduced by the noise we add into the system. And so the false detection rate here is just 84%. It's huge, right? And and if we go to the power of 10 to the minus 3, and we identify using the naive approach, we can record just 12 out of 19 significant SNP, but with just a false detection rate of 77%. And to the level of 10 to just minus 5, right? We got 9 out of 14 significant SNP with false detection rate of 70%. Then we're trying to improve this result, right? And my students present this afternoon, we use a hypothesis-based approach. Because the SNP-based approach is very pessimistic, right? We're assuming individual SNP is independent from each other. Well, human genomic data has structure, right? You can break the SNP sequence into those hypertype blocks, right? So SNP within one block, they're highly related. SNP between the blocks, their relation is is very loose. So we use this structure, we just add noise to the hypertype instead of individual SNP. So in this way, we can reduce the amount of noise added to the system and trying to preserve as much utility as possible, right? And so with an identification rate of 3%, which even lower than false positive, the result is we can get just 20 out of 22 significant SNP when the cutoff value in the significant threshold is 0.05. And then the false positive rate has been reduced from 84% to the 61%. It's still significant, but we're making progress toward this direction. And under 10 to minus 3, threshold, 10 to minus 3, just cutoff value, and we identify all 19 SNPs, significant SNPs. False positive rate, in the first time it is below 50%. So that means at least half of the SNP identified, they're true positive. And we need to go to 10 to minus 5, right? And in this case, we got we caught all 14 SNPs, right? False positive rate is just 47%. And then other group, we have University of Oklahoma, they got at the false detection, at the identification rate of 71%, which is a bit high. That means the noise that has been added there is insufficient to suppress, the identification rate. And so they got the they identify and they caught all the important SNP, but at a very big false detection rate. It's around 90%, 80%, right? University data, they got, just, and they also got all the important SNP. And, just, but first detection rate is 80%, 80%, 70%. The result is comparable, just, I think the detection rate is higher than baseline. And, just, and the false positive rate is comparable. And from, McGill, and they also got all the important SNPs. Except just a cutoff value of 10 to the minus 3, and again with a high false positive rate. But when we're moving on to a larger data set, more challenging data set, which contains 610 SNPs, and then just no kind of result, right? Just our hypothesis-based result no longer perform as good as we expect, and it is just we have 80% from 50% to 80% false positive rate. And even if we call the vast majority of significant SNP, and for the University of Oklahoma, and they also have just 95%, 90%, 87% false positive rate, just even if they caught all the just significant SNP, right? And the university data and their result is very good. They don't have false positive. They got all the accurate result. But just the algorithm we found, we discussed with them, and the algorithm is a little bit, just specific. They specialize this approach according to not only chi-square test, but according to the data set, right? The data set we gave. And just the McGill, they got most just significant SNP. But the false positive rate is still high, but it's way lower than other group except. Just university does. Of course, their approach is less specific. It's not special for all the data and the algorithm. So as we can see here, just and if we use, if we release the aggregate data, if we directly add noise to the aggregated data, right? It will work to some extent, just and in terms of, just oftentimes we can get all the important SNP. But the technique is still, in my opinion, far away from practice. And because, just looking at the false positive rate, right? We are talking about something, around 50% at very least, and oftentimes we can get 80%, 90%, right? That means you report a lot of important SNP, but only have, just a relatively small fraction of it. It's indeed important. So, just, and we are making progress on this end, but there's still some distance. By comparing the second approach, which is based on the idea that is we have a center, just like iDASH, they host the data, they don't release the data. But just we ask the research different, just the researcher, bioinformatics researcher to submit their algorithm, right? So iDASH run this algorithm on the data set, and then release the outcome to the researcher. Of course, you cannot just directly publicize the outcome because according to our previous research, even the outcome itself can be called information, right? So you need to add some noise into the outcome to make sure they are privacy preserving, but hopefully they're still useful to the research. And turn out to this approach, it works much better. So just and here again, just we use a very high privacy standard, which is called differential privacy. So differential privacy is a standard that it can guarantee the privacy protection independent of algorithm, whatever attack algorithm you use, independent of auxiliary information that no matter what kind of background information you have, we still can guarantee this level of privacy protection. It is originally a lot of people thought it's in practice, and it's very long to go. And but right now, if we apply this standard on the just the second scenario, that is we just release the outcome of computation analysis, right? And the result is surprisingly good. So let's look at what we got here. Just again, we use chi-square run on the real data set. We do this genome-wide associated study. So what happens is, and we just release, we just report to the user just the top 1, top 3, top 5 SNP, right? This kind of value. And we repeat the experiment by 1,000 times, and we got an average, right? Just for example, just if we got in one round we got just for top 5, if in one round we just got 3, we just copy the 3. Got 5, and we just take it as 5. Then we average what we got across these 1,000 rounds. So each time we cannot directly reach the result, we need to add noise. So there's a randomness in select the SNP. Instead of select the real important SNP, we just those group they use expansion mechanism to randomly choose some SNP according to a certain kind of distribution to ensure and the result has been released that does not leak out patient information except this this privacy budget which was set to 1. As you can see, the result is surprisingly well, and for UT Austin, right, this thing 1,000 times. If we just look at the top 1, always 100% they can get it. They always get the real result without leaking any information. And just CMU got it the vast majority of times. They got they got 0.98. The vast majority of times they always get the true result. And this happened for 5,000 people. But if we move into 100K people, it's a this in the scale of very realistic GY study. The result is the same. They can get the same result. And if we move to top 3, right, in the most cases they can get the top 3, right? And the result is surprisingly stable across 5,000 people and 100K people. And to top 5, again, vast majority cases you can got all these 5 important people without any mistake, and in the vast majority case, with a very stable result across the 500 people and 100 cases. And the performance degrade, just somehow after top 10, the top 10, top 15 to degrade. But if we just aim at just those most significant people, top 5 people, it looks like those existing privacy preserving techniques, which can achieve very high privacy standard and with a very minimal impact to data utility. And if we're just looking at a small set of people, they almost always get it. So what do we learn from these things? And we found if we want to provide aggregate the data directly, the technique we have today is still not there. We still cannot get there. And even if we have the best scientists in the field, even if we come out with the best algorithm we can imagine today, still we can introduce a huge amount of false positive. But if we do something just like a centralized analysis, just like, for example, using iDASH as a platform, right? Scientists submit algorithm and iDASH run algorithm for them and send out the result and voice to the result, right? And it looks like we can achieve very high privacy standard without undermining the utility of data most of the time, right? This is a very surprising result. And the technique in the second scenario is very much approaching to the practical use. So this is very good use. But on the other hand, I think this result should be taken with certain caveat, because and if we can release just aggregate the data, always aggregate data. That's the best scenario because we publish the data out there, and the user, the data user can run different kinds of arbitrary association tests on top of it, right? Which is a good thing. Well, if you use a centralized approach, then for each association algorithm submitted by the data user, we need to come up with a customized noise adding technique, right? For chi-squared, it works well. For g-test, their technique which also works well. But for other, customized association test, then we need a manual effort to analyze them to come up with specialized algorithm in order to preserve the utility and still, achieve a high privacy preserving standard. So that's why we got. So we need to acknowledge a bunch of students, right, who made a significant effort, and we also need to acknowledge, the grant by iDASH and also, our grant. Ultimately, we have to thank NIH to support this research. Okay, so that's our finding.
I was just curious whether you have any thought about the degree effect to figure out the top 5. Assuming that the amount of noise, the impact will be similar. Why there's such a dramatic dropping off? for the second approach.
Yeah, just and just I think this is based on the design of the algorithm. Just the algorithm. So basic idea is they cannot they got top 10, right? They cannot give the top 10 directly to you. They have to select the just a significance based on certain kind of distribution, a certain kind of distribution. This distribution is designed, is customized for different, just for different purpose and different team. Actually, Austin and CMU, they have a different, distribution. But only distribution has a unique feature. They are going to protect those most significant people. And for those relatively nice people, protection will be loose, right? And these are, I think just right after this talk, and the CMU group and just the Austin group, they cannot be here, but they delegated to us, they give us slides and they give us, just the authority to present their work here. So the students is gonna show you the detail of the algorithm. Other questions? Okay, so I think just and we are running out of budget a little bit, but just
yeah, we're running late, so let's take a 5-minute break.
5 minutes break.
Yeah, come back.
data challenge competition. And is it possible to see my slides?
Okay, that's right.
Okay. So I'm trying to complete the second task, which is to devise a potential private mechanism to select the k-most significant SNPs. So the setting is a case control study, as in normal GWAS study. The input data include, as we have seen, genotype data for the cases and minor allele frequencies for the controls. To rec the significance of the SNPs, we use the p-values corresponding to the Pearson's chi-square test association between SNPs and phenotype. To evaluate the performance of the algorithm, we use the define as utility the proportion of significant SNPs that can be recovered after the perturbation of the differentially private mechanism. So my method is based upon the exponential mechanism, which I'll explain what it is in the next slide. I'll talk about two variations of the method and the pros and cons of those methods. Of course, this is the slide with the most words. So differential privacy, as I assume many of you have known, is that given two databases that differ by one record, a randomized mechanism K is epsilon differentially private if the probability of releasing the same output using either database as input is bounded by e to the epsilon. And the sensitivity of a function is defined as using two databases that differ by only one record, how much the values of that function can change. So the exponential mechanism was proposed by McSherry and Tawar in 2007. Basically, in the context of this challenge, we can think of the database consists of data for m SNPs indexed from 1 to m. Then E epsilon is a random variable that has the following this probability mass function, which is proportional in log scale to a score function divided by the sensitivity of the score function. McSherry and Tawar shown in the paper, 2000 paper, that if a differentially private mechanism is defined this way, then it is epsilon is differentially private. So we can use any scoring function Q that uses the database's input for the exponential mechanism. In this presentation, I'll show two such scoring functions: the chi-square statistic and the Hamming distance, which was defined by, which was proposed by Johnson and Smatikoff in their 2013 paper. So the previous exponential mechanism will only allow us to select one SNP out of many SNPs. So in Johnson and Smatikoff 2013, they extended the exponential mechanism to allow us to select k significant SNPs. So the mechanism, it goes by basically repeatedly apply the exponential mechanism, but in each iteration, they will set the weight of sampling a SNP to be 0 if that SNP has been selected, and reweight the sampling scheme In Yu et al. 2014, we showed that the mechanism proposed by Johnson and Schmetikoff is also the epsilon differentially private. So I'll briefly talk about the performance of using different scoring functions. In the figure here, the x-axis denotes the privacy budget epsilon, and the y-axis is the utility. The solid line is for the chi-square statistic, when we use the chi-square statistic as the scoring function, and the dotted line is when we use the Hamming distance as the scoring function. We can see that for when epsilon is smaller, the Hamming distance approach always outperforms the chi-square statistic approach. However, after some big epsilon, we see that the utility of the Hamming distance function will tell before it reaches 1. I'll explain why that is in the after I give you more details about the scoring functions. I'll come back to this slide afterwards. To talk about the scoring functions, I want to talk about the assumptions that I've made in this problem. So the first assumption is that I assume that the number of cases and the number of controls are equal, and I to know that by n divided by 2, where n is the total number of individuals. I also assume that the case data are private, but the control data are known. I'll explain why that is necessary in the next few slides. In general, we can summarize a SNP by a genotype data in usual genome-wide association studies. But in this specific challenge, we only have a partial genotype table because we only have genotype data for the cases. However, we can derive a allelic table which only has alleles 0 and 1 in case and controls. And in this particular challenge, we actually use the chi-square statistics for the Pearson's chi-square statistic for the right allelic table to evaluate the importance, the significance of a SNP. So when we use the chi-square statistic as score, we know that the chi-square statistic is what we use to rank the significance of the SNPs. And as a result, high utility is always attainable by increasing epsilon. Let's look at this graph. The x-axis is the rank of the SNP, and the y-axis are the chi-square statistics sorted in descending order. We can see that the gaps between the top SNPs, which means that if the noise is small enough, we can quite confidently the algorithm will be quite confident to be able to select the top SNPs, the correct top SNPs. However, when the noise is big, then the noise when the noise is big, then perturbation of the mechanism of the algorithm will kind of demote the top SNP or promote the SNP that are not significant. So therefore, as we increase epsilon, the noise becomes smaller. We can always increase the utility. So when we use the chi-square statistic as the scoring function, one of the difficult things is to find the sensitivity of this chi-square statistic. Fortunately, in one of the papers, we have shown that that we can actually find the sensitivity of the chi-square statistic of an allelic table with positive margins and given number of control cases and controls. When we use the Hamming distance as score, which as all of you have seen previously when this organizer presented the result, this is the approach that's very powerful. So basically, when you're given a database and a threshold p-value, it will determine whether the database you have is significant or not. When the p-value of your database you have is smaller than the threshold p-value, then we define it as a significant database. Then we find a path from the present database you have to a database that is not significant, meaning that the p-value of the resulting database is bigger than the threshold p-value. Then we consider all those paths and find the shortest path and define that as the Hamming distance. So basically, the Hamming distance is in the space of all 2x2 contingency tables with positive margins. So when we use the Hamming distance as score, we define the score as positive only when the original database is significant, meaning that the original database has p-value smaller than the threshold p-value. One thing I want to note is that the significance of the SNPs ordered by these Hamming distance may differ from what we use to evaluate the synthetic SNPs, which meaning the chi-square Pearson's chi-square statistics. This can be problematic, which I'll explain in the next few slides. Also, this Hamming distance approach is sensitive to the choice of the threshold p-value. Also, because we have no genotype data for the controls, in order to use the Hamming distance approach, it becomes necessary to assume that the controls are known. So how do we find the Hamming distance? We can either use search all the paths exhaustively and find the shortest path, or we can follow the path of the greatest ascent or descent. One of the disadvantages of the greatest ascent descent approach is that we cannot always guarantee that we find the shortest path. The shortest ascent or descent approach is computationally tractable. So that's what I use for this challenge. So to do that, we need to find the gradient of the chi-square statistic so that we can do ascent or descent. In this particular case, because we have a partial genotype table, so we differentiate the chi-square statistic. We find partial derivatives with respect to the genotype table instead of the allelic table. Now let's go back to this graph. As I mentioned before, because when epsilon increases, we expect the noise to become smaller. However, because the Hamming distance approach does not always have result in ordering of the SNPs that are the same as the chi-square statistic approach, we may not be able to achieve really, really high utility when we increase epsilon. So this may become a concern if high utility is of interest. So to summarize, when we compare the both scoring functions, the computation of the chi-square statistic is really trivial because we have to calculate it anyway. But for the Hamming distance function, it can be really computationally expensive. On the other hand, finding the sensitivity of the chi-square statistic could be non-trivial. However, in some cases, we might be able to use upper bounds for the sensitivity. On the other hand, for the Hamming distance, the sensitivity is always 1, so we don't have to worry about that. I say the chi-square statistics approach, scoring function approach is stable because it is what we actually use to rank the significance of the SNPs However, for the Hamming distance, as I mentioned before, it is not always the case that scoring by Hamming distance will result, sorting the SNPs by Hamming distance will not always result in the correct ordering. So, in summary, I presented this exponential mechanism, which is called LOSIC by Johnson and Schmadikoff, talk about the patchwork statistics and the Hamming distance as scoring functions, and I show you a comparison of those two scoring functions and the performance of them. Thank you. So.
I have a question. You mentioned for the log-seq, you must assume the allele frequency of the control data is known.
Yes.
Why is that? Why is this
So, so you think about when you change one individual in your database, right?
Mm-hmm.
So in the allelic table, you have so 0, 1, 2. That's your controls. You have N1, N0, N1, N2. And in your allelic table, you have N0 plus N and N1 plus 2N2. Right? So, but in your allelic table, say you have A0 and A1. So therefore, N0 is equal to A0 minus N1 divided by 2. Right. Mm-hmm. So you can see that N0 and N2 are dependent on N1, but you don't know how many people with this genome type in your database. So if I say I change one individual in a database, I can keep subtracting, converting people with genotype 1 to genotype 0, genotype 2. But you don't actually know how many you have. What's the actual N1 here, right? So you don't actually really know what your database is.
But that's a Weinberg equivalent, right? If you know N0, N2, you know N1. Essentially, that's because
But we don't know the allele frequency. We assume that.
Okay.
Right.
Oh, we can discuss. Okay.
So, questions? Yes.
Yeah, so just at the very beginning of your talk, you compare these two approaches. And by the way, one approach is just an UT Austin's they also participated in the competition, and this is basically the state of art of just in this particular area. So at the very beginning of the presentation, you just mentioned one approach, one is perform better when you have a large just and privacy budget, right?
No, I was just saying that right here that when you increase epsilon, you kind of expect the utility to always increase until it reaches 1, right? But in this case, because the ordering of the UT Austin approach is not always so when you increase epsilon, your noise is really, really small. So you're basically outputting your data non-privately, right? But in this case, if you output your data non-privately, you won't be able to really output the real data set, the non like the real top case SNPs, even without.
Even for very high privacy budget,
yes.
be able to.
Oh, okay.
With really, really little noise, you can't output.
You can increase their just utility in the way that you expect. Right?
Yes.
Okay. Yeah. Just and this in terms of differential privacy, just it's indeed just and this is another good feature, right? Because just differential privacy has this nice feature that just and the privacy protection decrease gracefully and just utility also increase just gracefully, and this is very, I think it's very important feature in terms of differential privacy. But when we come to practical use, right? And in practical use, just rarely people use just and and privacy budget. If you use
Right, I understand that the privacy
about
budget can be
one
too big, right?
Yeah.
But my opinion is that we don't really know what epsilon means right now.
Oh yeah, of course.
So,
Of course.
right, so I think it could mean different things in different contexts.
But typically, just and if you have a budget about one, and people start worrying, right? Just because just and I remember Cynthia and mentioned use 0.5 is good, using 1 is okay, right? If you're using about that, it's become problematic.
Right, I understand that, but again, my opinion is that epsilon we don't fully understand epsilon at this moment. So, I mean, that's why I want to show you this graph, is that, I think if you can precisely define a attack problem, and then in that case, we can talk about what epsilon means, right? Yes.
So for this graph, we plot the utility is evaluated use chi-square, right?
Oh, I'm sorry.
You calculate the utility by chi-square testing.
Yes.
So the real reason is actually using Hamming distance. There's a discrepancy between the distance value and the real distance value.
Yes. So the ordering of the SNPs will be different if you use Hamming distance.
But that's expected, right? I mean, if you use a different scoring function, the order will be different, and that case you cannot preserve 100% of the order, even you have very high.
Exactly. But I just want to point out the nuance of using something else other than the actual scoring function that you used to.
Always
do. Yes, you always have some kind of things like that happening. Yes.
I was just wondering if you consider other summarizations of the contingency table in the chi-square statistic that might have not the same dynamic properties as the chi-square statistic?
Yes, like the ratio test, the G test, as was mentioned before. That could be one thing.
What do you think the benefit would be of having not such a huge dynamic range? At least.
Yeah, I think the approach is to do that case by case. So, I mean, like say in your study, you want to release the G test and as well as the chi-square test statistics, right? So you have to, output them, calculate them separately.
I was more thinking in terms of your sensitivity, because you get, in some sense, penalized by large sensitivity.
Yes.
So if you reduce the dynamic range a little bit, I don't know if the sensitivity of the, for instance, the odds ratio. And the odds ratio together with the marginals fully describes your contingency.
Mm-hmm.
So in some sense, it would substitute nicely for a chi-square statistic. And you would not have, I'm guessing, you wouldn't have the same utility problem, so convergence to when you get higher.
But the sensitivity is specific to a particular
I know.
function. So if you use the G test, it could happen that even though the sensitivity is smaller, the actual statistic is also smaller.
No, so it's a trade-off between the sensitivity and the magnitude change for the significance. I was just asking if you sort of thought about it.
Yeah, I did think about using different test statistics to do this kind of things. I was also thinking about 3x2 contingency tables. But I don't know. I'm not sure if that would change the result. And the Johnson-Schmadlikov approach uses something that's independent of the test statistic, which result in this great result. But sometimes if we can cope with the nuance of that approach.
Yeah, quick question. You mentioned when you do Hamming distance, there's two ways. One is the gradient descent, and the other is the exponential surge. which correspond to this figure.
Oh, so by descent and ascent, I mean that, so when a SNP is significant, then this chi-square statistic is smaller than the threshold chi-square statistic, so you do descent. So ascent meaning that if the original SNP is insignificant, then you do ascent until the chi-square statistic is greater than the threshold chi-square statistic.
Yeah.
Yes.
You had to assume n/2 because why? You didn't need
Because I think it's because I asked the data of the audience, but I actually it's also I think in the paper it's more general than that. You can assume any arbitrary number of cases and controls.
because you could assume what they gave you, right?
Yeah.
Would that be make it better or worse?
It could change the sensitivity of the function slightly, but I don't think it's that much a deal. It won't change the result that much. At least for the chi-square statistic, the sensitivity won't change much if the cases and controls are not that different. Which is like by a factor of 5.
Mm-hmm. So why did you make him do n/2? Or did you?
I guess it's just for convenience.
Okay.
Yeah. I don't remember any reason for that.
But we're gonna hold that number.
Okay.
I guess it was because only the allele frequencies for the controls are given.
Mm-hmm.
Right.
And so it's convenient to just match the.
We basically in the test we know roughly they have equal number case control. Probably that's the reason I over to.
Yeah.
Okay.
So.
So actually, just and the Johnson and the Smetcov, they cannot come here, and so we just present on their behalf. But the good thing is, and in CMU's talk, they already give you guys all the detail about just their technique. They actually compare their approach with Austin's approach. But anyway, we will run through their slides and give you guys some high-level idea of what's happening there.
Okay, thank you. Because just as Xiaopeng said, Aaron and Vitaly cannot attend this workshop, I will present his method and the results of test 2. And thank them for sharing the code and the slides. GWAS have become a popular method for analyzing genomic data in order to discover disease-related genetic markers. A typical study examines a large number of SNPs in a population of patients with a certain disease and a control population without disease. To do GWAS, datasets have to be shared between hospitals, biomedical research organizations, and other data owners. However, this sharing is not safe. So we propose a framework to solve this problem. The SNP stands for single nucleotide polymorphism. From the picture, we can see that DNA molecular 1 differs from DNA molecular 2 at a single base pair location. SNPs in the coding region are of 2 types, synonymous and non-synonymous SNPs. Synonymous SNPs do not affect the protein sequence, while non-synonymous SNPs change the amino acid in the protein sequence. So variations in the DNA sequence of humans can affect how humans develop the disease and respond to pathogens, chemicals, drugs, and other agents. With the phenomenal advance in DNA sequencing technologies, the cost of DNA sequencing is dropped dramatically, and more human genomic data becomes available. The objective of GWAS is to analyze genomic data, find the correlations between SNPs and disease. Usually, the number of SNPs in the output of GWAS is much smaller than the total number of SNPs involved in the study. Here's a diagram about case control GWAS. Researchers collect genomic data of a group of individuals with a certain disease and another group of individuals without. They then compare two sets of genomic data and find disease-related SNPs. In this example, SNPs on position 3 and 8 play a significant role. So many GWAS also report correlations among SNPs that are significantly correlated with the disease, but not correlated with each other, may represent independent risk factors for the disease and indicate the existence of different biological mechanisms associated with the disease. A study may report individual correlations between SNP pairs of interest or a heat map of correlations among all SNPs in a region. Given correlations, the statistical technique proposed by Homer can determine the presence of a patient in a case group by comparing her alleles with the allele frequencies of the case group and those of a reference genome. One's method can reconstruct raw DNA sequences. Given the SNP mechanism is differentially private if every output is produced with similar probability, whether of any given input is included or not. In this example, 6 record doesn't significantly affect the output distributions. The risk for C doesn't increase much if her data are included in the computation. In naive scenario, there is no well-designed GWAS queries. So data analysts have to query like this. They first ask, what's the correlation between two SNPs? And then they ask, what's the top 10 SNPs most correlated with the disease? All these are the output of the study. The analyst does not know them beforehand. So this framework provides 5 privacy-preserved GWAS queries. Number C returns the number of significant SNPs in the dataset with a factor 2 approximation about K. And the log C returns locations of top K SNPs from a specific block. So we mainly use log C in our computation. And there are other 3 queries, like log block returns the location of the longest correlation block. SNP p-value returns the p-value of a given SNP, and the SNP correlation. All 5 GWAS queries is privacy preserved. So using combinations of these privacy preserved GWAS queries, data analysts can perform a practically privacy preserved studies. So they first use, for example, they first use number-seq using GCAT to ask for the how many significant SNPs, and then they use the log-seq in our computation. get the SNP ID. And finally, they found the correlations. So a standard tool for designing differentially private algorithm for complex output spaces is the exponential mechanism. To apply this mechanism, one must define a score function that assigns a value to each possible input database and output pairs. In this framework, we define a general score function D called the distance score that works for arbitrary output spaces. It's based on the number of input modifications needed to change to or from a given query values. It's highly accurate. It's based on finally, the result, you may have seen results. The result is pretty good when k is smaller than 5 and acceptable when k is 10 when using large data sets. Thanks.
So basically, he just brings some background knowledge because in the CMU's talk, they already gave all the details. So he just gives some background knowledge about all the works, they've done with there. Just having distance-based, solving functions.
Question. So you mentioned about like back and forth for correlation and also differential expression. So when user expects to see the top 10 or 5 significant SNPs, are they all like independent? None of these are correlated, and then top, or even within the top, some of them might be correlated.
Yeah, I think
what is output?
In the simple scenario, we usually assume SNP is independent and to calculate the correlation between the SNPs and the disease. So when we use top 10 method to get the result, I think the result might be correlated. But when we calculate the SNP significance, we assume they are independent.
So that's the standard way of doing GWAS study, right? Of course, they are hashes much for more information about these things, and people are trying to use correlation to do GWAS study, but standard way is that they treat individual SNP to independent from each other, right? They calculate the associated study on individual SNP.
My question was the calculation correlation was purpose of looking at the correlation was to have the independent SNPs, or to take into account getting some correct result regardless of the parity. So that was my test. Initially, I thought was you wanted to get correlation of between SNP because if you say you have like 5 SNPs are correlated, you pick any of them, and you can just they all have to cut off in output. Right?
No, those basically the linkage discrete is not considered as an effective method.
So because the result without adding any noise, right? They calculate in the weight based on individual SNP. They're treating them as completely independent from each other. So you cannot directly release the top 10 result without any noise-based protection to the user because it leaks out information as we showed in the previous research. So what they did is they use exponential mechanism to randomly draw according to certain distribution on the system, to make sure the result is differentially private, but still, preserve most of the data. Yeah.
Okay, so we have 1.5 hours for lunch. I will be back at 1.
Back at 1, right?
Yeah.